vaccines, and also of a class of inhibitors that blocks SARS-CoV-2 entry into cells. There are two parts to my talk. The first part, I shall describe our efforts to construct what we call the landscape of antibody responses and integrate them with the model of SARS-CoV-2 infection dynamics within an infected individual, and use this framework to predict mechanistically the protection conferred by vaccines at the population level. Next, I shall present a mathematical model of SARS-CoV-2 entry into cells that helped us reveal an unexpected synergy between drugs that targets different pathways of viral entry. And we use this model to quantify the relative usage of different virus entry pathways by this SARS-CoV-2 virus. So before I begin, let me thank and acknowledge my collaborators, Rajat Desikan and Narendra Dixit. Rajat is at Cetara, a biosimulation company. Narendra is a professor at the Indian Institute of Science. So let me talk a bit more about our COVID-19 vaccine work. Let me show you a screenshot of current status of COVID-19 vaccines. As of December 13, we have 9 vaccines that are in full use. 16 are in limited use. About 112 are in clinical trials. And more than 100 candidates are in preclinical development. A major challenge, in my view, is to understand how to fast track these candidates into clinical use, thereby increasing the deployment of vaccines globally. Now, how do we do it? Here's an article that came just a month ago in Nature Medicine. The authors argue that if we have a correlate of protection of these vaccines, it will significantly decrease the drug development time or the vaccine development time. It might be even possible to skip or bypass the phase 3 clinical trials that evaluate the efficacies of vaccines. We kind of share this sentiment, and we have been looking for immune correlates of vaccines for a while now. Let me begin by showing you our analysis of data from phase 1, phase 2, and phase 3 clinical trials of 8 major approved vaccines. Shown here in y-axis is the protection conferred by these vaccines against symptomatic infection. Shown in the x-axis is what we called NT50 or neutralisation titers. These are basically quantified by drawing plasma from vaccinated individuals and evaluating its effectiveness in cell culture system. The larger the value, the more robust the antibody response is. Now, a quick short note, the antibodies are thought to bind to spike proteins on the viruses and prevent virus entry into target cells. There are other proposed mechanisms, but currently the thought is the neutralization is a dominant mode of action. Now, as soon as we accounted for study-to-study variability and estimated the scaled NT50, I won't go into the details, but as soon as we accounted for the study-to-study variability, the correlation between protections in clinical trials and the NT50 became more apparent, as you can see in this graph. This suggests that antibody responses might be a very good correlate of COVID-19 protection. Now, while this paper was in review, there's a couple of other studies that came up with similar correlations. They've used statistical models to explore this correlation further. What we instead did is asked whether we could mechanistically describe the correlation between protection and the antibody neutralization titers. In our view, to do this, we need a mechanistical framework that first describes the diversity of antibody responses within an individual and use this diversity description to describe COVID-19 vaccine efficacies. We developed a framework, a multiscale mathematical framework to do this. I shall present our model in the next few slides. The first thing that we did is quantify the properties of each antibodies. So over the past 2 years, many groups have isolated antibodies from patients infected with COVID and recovered, and quantify the properties of these antibodies. Shown here are 4 different representative antibodies. Shown in y-axis is the fraction of infection events affected by antibodies as a function of antibody concentration. Names of the antibodies are presented on the labels. Symbols are data taken from published studies. Lines are fit of a simple equation, 2-parameter equation. This equation has 2 parameters. One is IC50, the other is M. IC50 is nothing but the concentration at which the inhibition is 50%. M is the slope of the dose response curve. We can now transform this expression so that the fitting becomes more robust and the parameter estimation becomes much more easier. We make 2 key observations from this analysis. One is that the IC50 varies quite widely across antibodies, and also the slope also varies across these antibodies. We analyzed about 80 such antibodies from about 20 studies and collated this data and quantified these 2 metrics, M and the IC50. This simple model is able to nicely describe this data set. More importantly, what it allowed us to do is create the landscape of the antibody responses. Now, experimental studies have shown that individuals have a small subset of 5 to 10 effective antibodies in them to control the infection. So what we really hypothesized is each individual samples a small subset of antibodies from this large landscape that we created. So to test this hypothesis, what we did is looked into what's called a plasma dilution assay. So the assay works as follows. Here again, researchers draw plasma from convalescent patients who have recovered from infection, and then they dilute the plasma dilution. They dilute the plasma and look at the effectiveness to block infection. The curves look somewhat like this. Higher the dilution, higher the fraction of infection, uninfected cells that are blocked, which means the lower the effectiveness. Now that we have the landscape, what we did is we did some simulations. We randomly sampled 10 antibodies from this landscape, simulated these plasma dilution curves using 2 standard models in the field. One is Lowe, the other is Bliss. Lowe describes when antibodies act on the same sites, whereas Bliss is well suited when antibodies act at the different sites of the SARS-CoV-2 spike proteins. If you look at this expression, N is the number of antibodies used, DI is the concentration of each antibodies, IC50 and M comes from the landscape, and we have gamma, that is the dilution term, and the effectiveness of the combination that comes out of this expression. So what we can basically do is simulate the plasma dilution assays that experimentalists have measured and predict FU as a function of dilution. Here is the simulation of about 100 such patients shown in gray lines. FU, fraction unaffected by plasma dilution, as a function of different dilution concentrations. Colored lines are 3 different patients taken from this data set. This kind of suggests that our model is, or this framework is able to reasonably capture the plasma dilution assay. We wanted to further validate this. For that, what we did is we calculated what's called NT50, which is nothing but the dilution at which the inhibition is 50% at different antibody concentrations. Now, shown in these lines are predictions from Bliss and Lowe models, and the blue symbols are patient data taken from this study. Again, this framework is able to nicely capture the relationship between NT50 and antibody concentration, giving us confidence in the landscape that we have created and the way we sample antibodies to predict these dilution curves. So we think we are in a reasonable shape to try to predict the effectiveness of antibodies within infected or vaccinated individuals. Now, to relate to the efficacies of vaccine, what we need is a model that describes SARS-CoV-2 dynamics within infected individuals. We developed a framework for that. In this parsimonious model, we have target cells that get infected by viruses to produce infected cells. We have two compartments for infected cells to describe delays in the SARS-CoV-2 life cycle. And the productively infected cells now can produce new viruses. These new viruses go on to infect more cells. Now, infected cells die at enhanced rate. They can also trigger immune responses. The immune responses can now protect target cells from being infected. This is the parsimonious model that we consider. Now, one could write rate equations to describe each of these populations, each of this population. For example, the rate of change of target cells is determined by rate of infection by viruses and the rate at which the cells get protected by the immune response X. Here, epsilon is the effect due to vaccine. We have set to 0 in unvaccinated individuals. Likewise, we could describe all these subpopulations and predict time evolution of each of these variables in the model. Now, how do we estimate these parameters? So what we did is we looked at 3 different data sets, one from the US cohort, the other from German, and the third from, I think, I don't remember. So we compiled, the third one is from France, sorry. So we compiled such that this population consisted of individuals who had mild symptomatic infection, as well as severe infection that required hospitalization, so that there is equal representation of all kinds of infection that we see today with SARS-CoV-2 infection. First of all, our model is able to quantitatively describe this data. Each color represents different studies. These are the representative fits, and we analyzed about 60 patients. What the model predicts.
Can I ask a question in relation to the model?
Yes.
So on this scheme, so it's about the immune response.
Yes.
So you assume that target cells. So I was thinking that, isn't it possible? So there are different types of immune responses, obviously, but I was wondering if wouldn't be one effect of the immune response to reduce beta in some way, so kind of reduces the probability of. So kind of what is the refractory cell? So is it like assuming that the immune response turns some proportion of the target cells into some protected cells that cannot be?
That's correct, Zoltan. In a way, you could also model this. If you model that this immune response is blocking beta, as well as this, both structures would give pretty much similar readouts. But mechanistically, the way we think about is these are due to immune molecules that are released from infected cells, such as interferon. These molecules now trigger signaling cascades in target cells to make them refractory. A more detailed mechanistical framework of this description is available in our early work, but here we have kind of reduced it to quite an extent.
So what is the main mechanism? So is it, so the immune response leads to production of antibodies, right? And what are antibodies doing? They attach to the virus, right? Or
this is a description in unvaccinated individuals. antibodies are produced in unvaccinated infected individuals, but the time scales are much slower. It takes about a week or two after the infection to build up. So it's unlikely that antibody responses would have an impact on the initial infection dynamics that we are analyzing, and hence we have not incorporated the antibody responses.
Okay. So, I didn't know it's so delayed. So, and what about T cells? Do they?
T cells would come in. So first of all, this description of refractoriness, it describes the first line of defense, which is through immune molecules. T cells would come in, and it would pretty much affect the second phase or the decline phase of your infection. We have kind of assumed that is a constant and lumped it into this delta factor. Although in reality, it is likely to be a function of time.
So this is an early stage immune response, which is less efficient, but happens quickly after.
It happens quickly. That's correct. But the other aspects of immune response are lumped into these parameters with an assumption that it's time invariant, but it's unlikely to affect what we want to do in the later on stages. I can come back to it in a bit more detail at the end, does it answer your question? So,
yes, definitely. Thank you.
Now that we have a description of SARS-CoV-2 dynamics within an host, and now that we have a distribution of parameters that are estimated from 60 odd patients, we next asked, can we now predict what happens in a vaccinated individuals? Now, I showed you that we have a framework to sample antibodies in a vaccinated individuals. We now couple this framework to the SARS-CoV-2 dynamics model and then predicted viral load as a function of time. Now, stronger the antibody response, smaller is the peak viral load, and shorter is the infection duration. This is the prediction of the model, which is consistent with vaccinated animal studies. So, which again gives some confidence in our framework. Again, this is a parsimonious model. It's parsimonious because this data that is currently available to us. Now, with this framework, what we can now do is predict the peak viral load as a function of antibody concentration. Now, for a fixed concentration, you would notice that there is a large variability in the peak viral load. This is coming due to two factors. One is variability in the one is due to the diversity of the antibody response across individuals, and also the variations in the viral dynamics parameters. These two coupled together to give a large variation in the viral load for a given antibody titer. Now, the value 100 here is the detection limit, which is typically used in clinical trials to determine whether a person is protected or not. So in our simulation, any individual who has a peak viral load less than 100 is deemed protected. Now, one could now predict protection as a function of antibody concentration. Now, we did a large simulation sampling different concentration, different viral dynamics parameter, and then predicted, simulated the clinical trials. And now we predicted protection as a function of the scaled NT50 or the neutralization titer. So long story short, we have a framework that links phenomena happening at multiple length and time scales. And then this framework is now able to predict protection as a function of an immune correlate. Now, we are in the process of predicting protections for different. So this is, I must tell you that this is data with the original SARS-CoV-2 variant, not the mutant one. We are now in the process of predicting protections of all the new variants that are emerging. Now, it's time. Okay. We have also started developing frameworks to describe how antibody dynamics builds up post-vaccination. I'm going to skip this in the interest of time and switch gears and talk a bit about antivirals. So we recognize that protection is not 100%. Even against wild-type strain, some individuals get infected. This gets worse with new variants. The protection gets even lower even with the best vaccines that we have, such as Moderna and Pfizer. So we really need antivirals to combat this pandemics. So we have started to develop models of SARS-CoV-2 life cycle. Today, I'll focus on entry inhibitors for a few reasons. First of all, an early modeling study analyzing about 13 patients looked at different repurposed antivirals targeting the viral life cycles. They estimated the drug efficacies of these antivirals are between 20 to 70%. It's quite widely varying, but what they claimed is that this efficacy is much lower than what is required to confer protections or clear the infection by these kind of drugs. So monotherapies seem to be of low efficacy. One possibility is that we increase the drug concentrations of these drugs, but there are problems due to toxicities. So what we really need is combinations of drug treatments. And if these combinations are synergistic, then we might be able to achieve the desired efficacies at a much lower concentration with lower side effects. Now, we feel that inhibiting SARS-CoV-2 presents an avenue to realize synergy between drugs. Now, why is it? Here's a stripped down version of SARS-CoV-2 entry into target cells. To cut the story short, the spike proteins on the virus interacts with its receptor AS2 to enter into the cell. For this entry to happen, host proteases such as TMPRSS2 and Cathepsin has to cleave the spike protein. Now, TMPRSS2 is on the cell surface. So if TMPRSS2 is efficacious, the virus can just enter. If this is not, then cathepsin in the endosomes can cleave the spike proteins to let it enter the cell. So this creates two independent pathways accessible to SARS-CoV-2 entry. Now, what happens when you have drugs that blocks TMPRSS2 and Cathepsin? If we have a drug that blocks TMPRSS2, the infection will go through Cathepsin pathway, although at a much lower rate. Now, if you have a Cathepsin inhibitor, virus entry will happen through TMPRSS2 pathway. So what we really need is to block both the pathways simultaneously to achieve 100% blockade of infection. This would reflect as synergy with standard metrics used in the field. That's what we hypothesize, and we will test this hypothesis. We tested this hypothesis using a mathematical model, which I'll show in the next few slides. By the way, there are drugs that are targeting both of these proteins that are in preclinical development. Now, we considered cell culture assays because there's quite a lot of data studying this entry process in cell culture assays. Now, in these cell cultures, we know that TMPRSS2 and cathepsins are widely distributed. They vary across cells. The expression levels vary across cells. Because of that, the efficacy experienced by these cells also vary. For example, a cell that expresses low TMPRSS2 will experience a very high TMPRSS2 inhibition, whereas cells with high TMPRSS2 will experience a very low inhibition. This is true also for cathepsin inhibitors. Now, we developed a model describing these processes. First of all, we assumed TMPRSS2 is log normally distributed across cells. And then the susceptibility to infection or the probability of infection through TMPRSS2 is a sigmoidal function of the expression. Now, one could construct a viral dynamics model mimicking the cell culture assays. Here, target cells express different amounts of TMPRSS2. As we go from 1 to K, the cells proliferate, they die, they get infected when they are contacted by viruses. But the infection rate now depends on the TMPRSS2 expression. This is where the single cell level description couples to the population level description. As the TMPRSS2 increases, the susceptibility increases and the infection rate increases. Now the infected cells die, they produce new viruses, and the new variants get cleared. So this is the formalism that we have at hand. Now, what happens when you have drug? Drugs would reduce the TMPRSS2 levels in a cell, and it will reduce the amount such that the probability of entry to TMPRSS2 pathway will decrease, which can be nicely described by the set of phenomenological expression. By the way, in our past few studies, we have developed a detailed reaction diffusion kind of formalism. Those studies suggest that these are very good, reasonable assumptions to make. Now, here's a metric that is experimentally measurable, that is fraction of infection unaffected by drug, here the TMPRSS2, which is basically summation over different subpopulation of infected cell in the presence of drug divided by infected cells in the absence of drug. This is an experimentally measurable quantity. In reality, we have a distribution of both TMPRSS2 and Cathepsin, which is what we have in our model. So it's a joint distribution of TMPRSS and Cathepsin expression. And the probability also is a joint distribution, which is related in this form. The total probability is summation of individual probability minus the product. Now, here's a metric that quantifies synergy. Now, when both drugs are present, we can now predict fraction of events, infection events, unaffected by both these drugs. And we can also predict, calculate what is the expected fraction unaffected. If the pathways are acting independent, the expectation is that the fraction of the combination is just the product of fraction of monotherapy. So that's the term. So the difference gives synergy term bliss, which can vary between 0 to 1. A value of 0.2 or greater is it represents strong synergy. Now, we did quick simulations with mono treatments or dual treatments, and this is what we find. As the concentration of TMPRSS2 inhibitor increases, the fraction of uninfected events drops, and then it plateaus. It plateaus to a value 0.78 in our simulations, which means, so this is with Cathepsin inhibitor, which means even at the very high concentration of Cathepsin inhibitor, 78% of cells get infected through TMPRSS2. Now, when we do this with TMPRSS2 inhibitor at saturating concentration, 39% of cells get infected through the TMPRSS2 pathway. Now, a dashed line indicates when the additivity that's expected from this expression. The solid blue line is the prediction of our model. So it is quite intuitive. If both the pathways are completely blocked, the infection is also completely blocked. That's the prediction. But the dashed line is expected from standard metrics of bliss. So this difference between the dashed line and the solid line gives this vertical synergy, or what we call in this field a bliss synergy. So the expected value is about 0.3, but we get about complete infection. The synergy is about 0.3. Now, we did this simulation over a range of concentrations of TMPRSS2 inhibitor and Cathepsin inhibitor. Here's the expected value. Here's the predicted value. Here's the predicted synergy. As you can see, the synergy increases as a function of drug concentration. We have maximal synergy when both drugs are at maximal concentration, which means when both pathways are maximally inhibited, you have maximum synergy. Now, the question is are there experimental evidence to these predictions? Fortunately for us, there have been experiments performed with pseudoviruses harboring SARS-CoV-2 spike proteins or SARS-CoV spike proteins across cell lines with monotherapy and combination treatments. These are cell culture studies. It's either with Camostat, a TMPRSS2 inhibitor, or different types of Cathepsin inhibitor. Now, the experiments also measured FU in the presence of each drug. And it's straightforward to calculate what's the expected value when there's a combination treatment. The experiments have also measured the effect when you have a combination treatments. So the difference gives the extent of synergy that is observed in the cell culture experiment. As you can see, there's quite a bit of synergy that's observed in these experiments, which are now consistent with our model predictions. Now, the question that we asked is, what are the origins of the synergy? I think I'm running a bit late, so I'm going to move a bit faster. To cut a long story short, there's synergy at two levels. One is single cell level, and the other is a bit more involved, which is population level. So to do this, we made the expression level uniform in our simulations. All cells express similar level of TMPRSS2 and Cathepsin. Here again, we see synergy. The reason is, if you block TMPRSS2 pathway, the virus enters to Cathepsin, and vice versa. When you block both, there is synergy at a single cell level. We derived analytical expression, which helped us to explain this in a bit more nicer way. The Bliss synergy comes through this expression, and we could now quantify the Bliss synergy at the single cell level, depending on the probabilities of entry. Now, there's another origin of synergy. Consider a case where you have cells that express only TMPRSS2 and cells that express only Cathepsin. Now, if you have a TMPRSS blocker, cells with Cathepsin will get readily infected. And if you have a Cathepsin blocker, you'll have cells with TMPRSS that are readily infected. Now, only when you have this combination, the whole protection gets protected. This is what we call population level synergy. To elucidate this population level synergy, it was a bit tricky. So we derived an analytical expression, which is a function of probabilities, drug concentration, and the phi function, which describes the joint distribution. With this, we can now predict synergy, the overall synergy in black, and synergy from population level effects in blue, in red. And in this parameter settings, 43% contribution came from population level synergy, and rest to single cell level synergy. Next, and now when does these two types of synergy play out? That's the question that we asked. From our model, we could nicely work out a way to compute relative usage of different pathways. It nicely turns out to be a function of FUs in the presence of individual drugs. Notice that these are experimentally measurable values. This allowed us to quantify relative usage of the two entry pathways across different cell lines. Cyan is Cathepsin pathway, and TMPRSS2 is in blue. As you can see, some cells predominantly allow Cathepsin pathway, some cells predominantly allow TMPRSS2 pathway, some cells have pretty much similar usage of these two pathways. In these cell lines, there will be single cell level synergies. And what this data shows is that there is a spectrum of usage of these pathways across cell lines. We expect that this would manifest as different organs expressing different levels of these protease inhibitors, and this might have implications on how synergy might happen in vivo. So here's what, these are basically a summary of what I just mentioned. So with this, let me quickly summarize my talk. So first of all, we developed a model that linked different phenomena and predicted the population level protection conferred by different vaccines. And then I described a model that elucidated synergy between two inhibitors that block different steps of virus entry. We also calculated the relative usage of different pathways. With this, I'll stop. And if there's time, I would be happy to take questions. Thank you.
Thanks, Pranesh. It's a great talk. So yeah, we can take like 5 minutes if there are any questions.
Mm-hmm.
There are some questions in the chat, but let's wait if there is.
So there is some.
Yeah, the first question is asking about the drugs that attack, the name of the drugs that attack both pathways for the disease.
Yes, we do have we do have some candidates that are in clinical development. Here are these drugs that block TMPRSS2, and here are some of the drugs that blocks cathepsin. Inhibitors of TMPRSS2 is much more advanced, but those of cathepsin, I think, are much more in preclinical testing. And there was a clinical trial that kind of tried to test the combination treatments of TMPRSS2 and cathepsin inhibitors. Unfortunately, I think the clinical trial didn't go ahead. I haven't followed it up very well. But there's a company in India, which is called Sun Pharma, and I think they might be trying one of these drugs, but I'm not 100% sure.
All right. Okay. Maybe any other question? Okay, second question from the chat. Mm-hmm. It says, why do people say the low viral load doesn't lead to disease?
So it is a great question. The relationship between viral load and the disease outcomes are not clear. As you know, the disease outcomes are quite heterogeneous, symptomatic, mild and symptomatic. And some have very severe infection. The issue is the link between viral load and how the immune response are dysregulated is not clear in my view, and hence the link between viral load and immune-mediated pathogenesis is not clear. Notice that in SARS-CoV-2 case, it's not the virus. Virus gets cleared. It's mainly the dysfunction of immune system that's a problem. So linking these two, it's a challenge, and we are actually working on this problem, trying to tease apart how different arms of immune response contributes to diversity of infection outcome. This is something this is a work in progress.
The second part of the question says, can your model calculate the threshold viral load below which disease expresses no symptom?
Again, it's a great question and a tricky question. Unfortunately, I don't know the answer, and I also feel the link between viral load and symptoms, it's not very clear. It might be understood by others, but at least to me, it's not very clear. How does viral load affect different symptoms? Might also depend on host factors. So that's where I think my understanding stops.
All right. Is there any other question? Okay, maybe the last question from the chat again. Mm-hmm. It says that, is it possible to model the effect on onset and progression of disease by the use of antiviral replication, therapeutic drugs such as, it's a long name for the drug, I haven't, hydroxychloroquine. Yeah, sorry about that. And there is another name for the drug. Mm-hmm. There are two drugs there, yeah.
It is possible, and I think that there are few groups attempting to do it. They might have even done it in animal studies. So I think the data that would be needed is administer these drugs and measure the viral load in placebo and in treatment case. Now we will get measurements of viral load. Now one could fit the viral load, the viral dynamics model, and try to estimate the efficacies of these drugs. Which is, if my screen is visible, this is exactly what the study did for different repurposed drug. What I think might be happening is the timing of treatment and the efficacies of these treatments might be very maybe determining factor. Okay. And although they might have effect that might not reflect in the viral load changes and so on. Yeah. So yeah. That's what I think about this question.
All right. Okay. Any other question? If not, let's say thanks very much again, Pranesh, and participants for joining us, and looking forward to seeing you in our next seminar. Thank you, Pranesh.
Thank you very much. Thanks
Yeah.
all.
Yeah.
Thank you.